In this lecture, I am going to go over the steps in meiosis. We've already talked about why meiosis happens and its role in sexual reproduction. So here really I'm just going to focus on the stages. On the screen, what you can see at the top are the steps within meiosis. In this lecture, I am going to talk about the stages in meiosis. We already talked in class about why meiosis occurs, and so my interest here is more just going over what happens during meiosis I, the first half, and meiosis II, the second half. What you can see on the slide is a comparison of meiosis and mitosis. So just to remind you, when we talked about mitosis, there was only one series of events. We had prophase happen once, Prometaphase, Metaphase, Anaphase, and Telophase, but those processes did not repeat. And as a result, we had only 2 daughter cells form during mitosis. What we will see in meiosis is that each of those stages I just mentioned happens twice. So for example, up here we see prophase, prometaphase, metaphase, anaphase, and telophase. That results in 2 cells, and then each of those daughter cells repeats the process, and so in the end we end up with 4 daughter cells in meiosis compared to 2 in mitosis. So we will talk about the stages in meiosis I first, and meiosis I is the more complicated part. Meiosis II is going to be very similar to what you have already seen for the process of mitosis. Okay, so within meiosis I, remember that this is preceded by normal interphase, just like in mitosis. We have gap 1, we have synthesis of new DNA, and we have gap 2. At this point, meiosis can begin. When meiosis begins, remember that we have sister chromatids that are genetically identical to one another, and they are attached in the middle or the centromere of the chromosome. And they're going to remain attached to each other throughout the entire process of meiosis I. The first step in meiosis is prophase, just as it is in mitosis. However, what happens in prophase of meiosis is different. Specifically, in prophase I of meiosis, 2 homologous chromosomes, or homologs, pair with each other. So one copy is inherited from the father, one copy is inherited from the mother, and these chromosomes have the same series of genes in the same sequence, but they may have different alleles for those genes. And you can see the centromere and kinetochore labeled. You can see the synaptonemal complex, so the proteins that hold these 2 chromosomes together. And you can see that each chromosome still has its 2 sister chromatids. And as we've already talked about, during prophase, we have the process of crossing over. That is when chromosome arms can break off and reattach to the other chromosome such that we have new combinations of genes. The points where crossing over occurs are called chiasmata, and we'll see a slide for that in just a second. And you should remember that this structure with the 2 chromosomes can either be called a tetrad because there are 4 there are 4 sister chromatids here, or it may be called a bivalent because there are 2 chromosomes here. So tetra referring to 4, bi referring to 2. And these are simply synonyms for one another. During prophase, chromosomes condense. This is just like in mitosis. The nuclear envelope breaks down. That is just like mitosis. The spindle forms, and the homologous chromosomes pair into tetrads or bivalents, as shown here. So now let's look a little bit more closely at what happens with crossing over and chiasmata. So here are two homologous chromosomes. You can see the A's and little a's, B's and little b's, etc. represent alleles or different forms of the same gene along the chromosomes. So you can see that these are in the same order. You can see here that there's a point where the two chromosomes cross over, and it's being shown schematically in a way that makes it really easy to see. So this point where they cross is a chiasma, the point of crossing over. And you can see that the result of this is going to be that after crossing over, the blue chromosome now has a piece of the red, and vice versa. We can also say that the entire purpose of crossing over is to make new genetic combinations. So if we look back here before crossing over, these two sister chromatids were identical, and they both had big A, big B, and big C. So they had the same variants of these three genes. Meanwhile, this other chromosome, both sister chromatids had little a, little b, and little c. However, after crossing over has finished, you can see now this chromosome, this sister chromatid on the left, still has big A, big B, and big C, but now we have a new genetic combination. Specifically, we now have big A, big B, and little c all on the same sister chromatid. So that's new. And on this sister chromatid, for the other homolog, we now have little a, little b, and big C. Again, a new genetic combination. So pairing of homologs and crossing over are the events that make prophase I in meiosis very different than prophase II in meiosis, and also very different than mitosis. What we'll see next seems a little bit more similar. In prometaphase, we have spindle fibers that are made of microtubules, and they are going to attach to the kinetochore proteins at the centromeres of the chromosomes. So those were these proteins shown in yellow right here and right here. And that will allow then the spindle to move these chromosomes towards the metaphase plate. At this point, the homologs are still held together. So homologs are moving as a pair, and what's going to allow them to move as a pair are the chiasmata, those points of crossing over. So for example, on this diagram, this point right here, there are actually proteins that will hold these two chromosomes together. So when this one moves, this one will be moving with it. When they move, it probably looks a little bit more like this, where they're still going to be tightly paired with one another. And these chromosomes will move towards the metaphase plate, which your book refers to as the equator of the cell. So finally, the pairs of homologs reach the equator, or the metaphase plate, and you can see that they are aligned. However, unlike mitosis, individual chromosomes are not aligning. Instead, pairs of homologs are aligning. So this is one pair, this is another pair in this example cell. And it is random whether the maternally inherited or paternally inherited homolog lines up on the left or right. So in this example, if we assume that red is maternal and blue is paternal, then for this top pair of homologs, the maternal chromosome is on the left, the paternal is on the right. However, for the second pair of chromosome, the opposite is true. If this exact same cell were to go through meiosis again, there might be a different arrangement. For example, both maternal ones might be on the left and both paternal on the right. And so this is referred to as random orientation of homologs in metaphase I. And this is going to be important to increasing genetic diversity among offspring, because in an individual, there will be many cells going through meiosis, and each cell will have a different random orientation of their chromosomes. This means that daughter cells will have genetic variation, and if that is true, then the offspring, when gametes from different individuals come together, will also have genetic variation. So we've now talked about the two main mechanisms that cause genetic variation to occur during the process of meiosis. To review, those are first, crossing over, and second, it is random alignment of homologs on the metaphase plate in metaphase I. So let's move on and see what happens in anaphase. Remember, homologs are still attached, or rather sister chromatids are still attached to one another. You can see that here, and then also here, and on these other two chromosomes. That's why there are 4 arms instead of just 2 on each of these chromosomes. But now at this stage, the homologs are going to break apart. So one homolog will be pulled in one direction, the other homolog is pulled in the other direction. And the chiasmata, those points of crossing over, are broken, which is what allows this separation to occur. So sister chromatids still attached, but homologs are now separate. That is anaphase 1. Telophase I is going to vary a little bit. Sometimes we will get reformation of the nuclear envelope around each of these 2 new nuclei. Sometimes that nuclear envelope will not reform and the cell will go on to prophase II. Cytokinesis will occur. So we will go from having one cell, and you can see the cleavage furrow forming here. And so this will separate into 2 different cells. These cells that have now formed are going to be considered haploid cells once cytokinesis occurs. They are haploid because we still have 2 sister chromatids comprising each chromosome, but now these are identical sister chromatids to each other. And when we have 2 sister chromatids that are attached to one another, we still refer to that structure as a chromosome. So in effect at this point, we've gone from having a cell with 2 pairs of chromosomes to having a cell with 2 chromosomes and another cell with 2 chromosomes. We've reduced the number of chromosomes by half. Now what we need to talk about is meiosis II. And the short version of meiosis II in a sentence is meiosis II is identical to mitosis. The only difference is that meiosis II is preceded by meiosis I, while mitosis is preceded by replication of DNA in the S stage. So notice that between meiosis I and meiosis II, there is no additional replication of DNA. All we're doing is we're going from dividing the DNA once to dividing the DNA a second time. All of the steps of meiosis II are shown here, and there's a description at the right, but we will look at them pretty quickly, stage by stage. So in prophase II, there's going to be some species-specific differences, but we're not going to be too concerned about these. Basically, if the nuclear envelope had reformed during telophase I, it needs to break down again. If it hadn't ever reformed, then prophase II can happen without this nuclear envelope breaking down again. Similarly, if the chromosomes had decondensed in telophase I of meiosis, then they would need to recondense into meiotic chromosomes during prophase II. But if they stayed in this condensed form at the end of telophase I, then they could simply go on and continue the process of meiosis without needing to recondense. And this is not a difference that I'm especially concerned about. So for our purposes, it's easiest to assume that the chromosomes just stayed condensed the whole time, and that the nuclear envelope did not reform. And that, I think, will give us a good general understanding of the process. Centrosomes, they will need to duplicate again. We go from 1 to having 2 centrosomes, and we are going to have a new direction for this meiotic event. And so these centrosomes are going to need to move to opposite ends of this cell so the process can move forward. So you'll notice here that the steps are pretty similar to the steps of prophase in mitosis. Prometaphase, just like in mitosis, is when we have the spindle become fully formed and microtubules from the spindle are going to attach to both sister chromatids. So we have a spindle fiber attaching to one sister chromatid on the left, and we have a spindle fiber from the other pole attaching to the other sister chromatid on the right. This is going to eventually allow these two sister chromatids to be pulled apart in anaphase, but we'll get there in just a minute. So this is very different than what happened in metaphase 1. I'm going to go back to metaphase 1 to remind you of this. In metaphase 1, it was a homolog that was attached to one pole, and the other homolog was attached to the other pole. But each homolog out of a pair is only attached to one pole in metaphase 1. Now let's go back and compare that to metaphase, or to prometaphase 2. In prometaphase 2, each sister chromatid is attached to an opposite pole, which means that each homolog is attached to both poles. Half of it's attached to the left, half of it is attached to the right. So let's see. Here's a diagram that shows this fairly nicely. So on the top half, we see what prometaphase 1 and anaphase 1 looks like in meiosis. On the bottom half, we see what prometaphase 2 and anaphase 2 looks like. And so it shows really clearly that in prometaphase 1, this chromosome is attached only to the left-hand pole. This chromosome is attached only to the right-hand pole, allowing these two homologs to separate. In meiosis 2, it's a sister chromatid attached to the left and the right pole, allowing the sister chromatids to separate in anaphase 2. Let's move on and look at metaphase 2 and anaphase 2. The events here are going to be the same as in mitosis. Sister chromatids are going to be pulled apart and towards opposite poles. Finally, we can look at Telophase II and Cytokinesis. The chromosomes are going to reach the poles and they are going to decondense. They are going to go back into their more spread out version, so they will not be individually visible under a microscope. We will definitely have reformation of the nuclear envelope, so forming new nuclei in both daughter cells. And we will need to have cytokinesis follow, dividing this one cell into two cells. Again, you can see the start of a cleavage furrow forming here. Let's quickly compare meiosis to mitosis. Both processes have in common that they divide nuclei and they both occur in eukaryotic cells. However, there are several differences. Mitosis has one division, meiosis has 2 rounds of division. In meiosis, homologs pair to form tetrads, but this never happens in mitosis. Next, in meiosis, we have 2 events that create genetic variation among daughter cells. These are specifically crossing over in prophase 1 and random alignment of chromosomes in metaphase 1. And note that I updated the wording. There was sort of more confusing wording on the version of this PowerPoint that I put online. And these two differences, create genetic variation, which is going to be important for sexually reproducing offspring. The goal of mitosis, in contrast, is to produce genetically identical clones, which is why neither of these two processes occur. Also, because we have 2 rounds of division in meiosis, it reduces diploidy. Organisms go from being diploid, or 2N, to producing cells that are haploid, or 1N. In mitosis, this does not occur. In animal life cycles that you are familiar with, it is diploid cells going through mitosis, and those diploid cells produce more diploid cells. And so I'm going to end just by summing up important points for the process of meiosis. First, remember that this is happening because sexual reproduction requires reducing chromosome numbers by half, such that fertilization can restore the original number of chromosomes. Remember that for meiosis, just like mitosis, it is preceded by a round of DNA synthesis. However, unlike mitosis, it has 2 rounds of cell division, not 1. And in meiosis, in the first round, it is all about separation of homologs. In the second round, it is all about separation of sister chromatids. And finally, as we've already said a couple of times, we have 2 processes, crossing over and independent assortment of chromosomes in metaphase I, that will create variation among daughter cells. And this will be important to allowing new genetic combinations in offspring.